Hi. Methods discussed so far about in situ and ex situ conservation can be used for plants and animals. But conservation of microorganisms need different approach. In this video, I am going to talk about conservation strategies which can be used for the conservation of microorganisms. Microbial culture collections are the preservation of microorganisms ex situ. There are no formal methods for conserving microorganisms in situ because all in situ methods are conserving the whole geographical area. So along with the plants, animals and its habitat, microorganism will be preserved automatically. But documentation of the microorganism population is very difficult because it is very diverse and very difficult to identify its presence. And so ex situ method of preservation of microorganism is not prevalent. So for that, the whole technique we are using for ex situ conservation of microorganism is coming under microbial culture collections. It was first established by Professor Franklin Gall in 1819. It involved the microbial culture collection. What they are doing is the acquisition, preservation, and the distribution of viable cultures of microorganisms. So they will collect the microbial samples, microbial population from different parts and from different peoples, and they will preserve it using different techniques. And once and when it is needed, they will distribute it to the researchers and farmers, industries, and everything. This microbial cultural collection preserve prokaryotes, which includes bacteria and archaebacteria, then virus, virions, fungus, algae, and protozoans. So all type of microorganisms can be preserved this way. These are the repositories for the type strains also, because each and every microorganism will have the substrains like Escherichia coli has the K strain, R strain, B strain, etc. So all the type strains will be also conserved. And these microbial culture collections are run by government, some cases some semi-government, private, university or industry. So different organization can run microbial culture collections. Hello. Roles of microbial culture collection. It collects, maintain and dispatch microbial cultures. They have the complete documentation and cataloging of microorganism. They are the source of authentic cultures, but when in contrast to the animals and plants, getting authentic cultures in microorganism is a little bit difficult because they are very small creatures and most of them they are looking alike. So taking a pure sample or pure culture is difficult. So this culture collections acting as a source for authentic cultures. It acts as a safe repository for microorganisms with restricted distribution. Some economically important microorganisms can be conserved this way. It provides identification services because if you want to identify microorganisms, they will help. It serves as a repository for valuable cultures. Things who have the industrial or the scientific values will be conserved here. It helps in education and training like any other in situ and ex situ conservation method. It promotes research and development in taxonomy as well as preservation. Authentication is the one of the main role of culture collections because these microorganism getting authentic cultures of a microorganism or a particular strain of microorganism is difficult. So they are doing it by following a set of practices which include if you are receiving a named cultures, they will confirm its identity. If you want to deposit one particular culture and you know what the culture you have, even if you are telling the name and the scientific name and everything, they will double check with their own expertise. Sometimes the cultures will be unidentified, you don't know what you are going to store. In those cases, they will be specialists checking the culture before incorporation to make sure the safety of the employees and everything. They will have some checking. They will establish molecular barcoding. The DNA level analysis will be done. The cysteine RNA sequencing and everything will be done to make sure the identity or identify the microorganism. And they will be just like the plant in any other in vitro culture methods, they will be regenerating the microorganisms in constant intervals, and we'll compare the barcodes with the previous one. At appropriate regular intervals, cultures are recovered and represerved. Then they protect intellectual property rights of the depositor. Whoever is depositing the culture, they own its rights. And proper documentation is one of the important parameter. So for documentation, they will be recording so many features of this microorganism, like from where it is isolated, what are the substrate or host of the microorganism, then date of isolation, name of isolation as an isolating, who is depositor, whether it's an institution or a company, name of the person identifying the strain and his qualification, and how he's identifying the strain, preservation procedures used, optimal growth media and temperature so that we can regenerate, date of biochemical or other data on biochemical or other characteristics, then regulatory conditions applying. So this all data will be collected and documented. And in every culture collections, the microorganisms are divided into risk groups. They have a regulation from WHO to divide microorganisms into 4 different risk groups based on its potential health hazard and pathogenicity. The risk group 1 are the microorganisms, they don't have any known risk, they don't cause diseases. Risk group 2, they cause diseases, mild diseases, but we have prophylaxis and treatment. We already have the vaccines or the antibiotics to treat the disease, so that is the risk group 2. Risk group 3, they cause severe human disease, but we have vaccines or treatment. So even if we got the disease, we can treat, or we can give the vaccines for the workers. And risk group 4, those are the most dangerous microorganisms. They cause severe human diseases and spread in community. Now you are very familiar with the community spread. So the risk 4 organisms has high potential of community spread, and it causes severe disease, and no effective prophylaxis or treatment is available. So there is no vaccine, no treatment, just like corona. They are the risk group 4. So first, if you take in a microorganism to the culture collection, they will first identify the organism and place it in one risk group. Based on the risk group, there will be aseptic conditions and containment conditions will be used. Preservation. The preservation method should ensure optimal viability, storage and purity of the microorganism. But the thing is that different microorganisms are different in their behavior, so they need different techniques for preservation. One technique which works for one microorganism may not work for another microorganism. So for each microorganism, we should optimize the procedure. Each strain should be maintained by at least 2 different procedures, ideally in 2 different locations. Then optimal protocols for preservation should be identified and followed. Proper documentation is made at all stages of operation. And basically we have 2 different methods of preservation for microorganism. Methods to keep the organism in metabolically active stage, and methods where organisms are submerged in submerged metabolic state. That's a suspended metabolic state. So in metabolically active state, microorganism can be preserved by periodic transfer to fresh media that I already told about the plant cells and animal cells, or overlaying the culture with the mineral oil. In laboratory, you might have done the culturing of microorganism in slant, test tube slant, so agar slants actually. So what we do after making the agar slant and after incubation, we will put mineral oils over the surface of the agar so that the oxygen exchange will be blocked. That will lead to reduce the growth rate. So the microorganism will be viable for a long time. That's the technique. Storage in sterile soil, which is used for spore forming microorganism like fungus. They will make spores. These spores are very resistant. They can be stored in soil, sterile soil. Saline suspension culture, also applicable for only few classes of microorganism. By adjusting the osmotic pressure, we can actually store. So these methods are actually the metabolically active stages are short-term storage methods, and we have long-term storage methods like submerged metabolic stage storage, which including drying in the vacuum to reduce the moisture content and oxygen level. Lyophilization is a freeze drying, then using like liquid nitrogen, ultra low temperature, stored in silica gel. So silica gels are used to reduce the relative humidity. So these are the methods we can be followed in the microbial culture collection. There are different microbial culture collections in the world. These are organizations will provide cultures for use. Most famous one are ATCC, American Type Culture Collection, USA, or NCIB, National Collection of Industrial Bacteria. So the industrially important microorganism for fermentation can be stored there. In India, we have MTCC, Microbial Type Culture Collection and Gene Bank, or MCC, Microbial Culture Collection. NAMCC, National Agriculturally Important Microbial Culture Collections. In the first one, ATCC, they are having microorganism as well as plant and tissue culture. So they act as a gene bank for plant, animals, and microorganism. But other culture collections, they only deal with the microorganisms. And some of them are very specialized, like National Collection of Industrial Bacteria or agriculturally important bacteria. But MCC is microbial culture collection. They have microorganisms, so they will vary. The microbial type cultures will have different strains of microorganism. This is also one of the largest in India. So getting the microbial culture from the culture collection is very easy. Here I will show how to purchase cultures, microbial cultures, or get microbial cultures from this ATCC. So just go to the Google and type ATCC. It is just like purchasing something from the Flipkart. ATCC's website. Go. Then this is the website. They listed the products. They have cells and the microorganisms. As I said before, they are selling all type of cells, plants, animals, and microbial cells. I am searching for Saccharomyces cerevisiae, which is one of the most industrially important microorganism. All the alcohols, breads, wines, and everything is made by Saccharomyces cerevisiae. Then these are the properties. Just like you are browsing in the Flipkart, you can see the name and description. Then yeah, the price is also listed there. If you want to know more, just click. Yes, here we go. So the detailed description, so-called specification, then characteristics. So these are the proper documentations are given for everything. Culturing method: if you get the organism, you should culture this way. History: who made it, how it is made. Just give the quality, add to the cart. That's it. Just like an online purchase. So in this way, we can actually assess the microbial culture collections. Unlike the in situ methods, you cannot actually buy animals like this. But for microorganism, it is easy. But they will have some restrictions. If you want to have a microorganism which is high risk group, like risk group 4, you don't get, because you need the letters from the government and everything to get such type of microorganism. Otherwise, the normal microorganism you can actually buy from this culture collections. World Federation of Culture Collections is actually a union, just like the union for everyone. They have a union for culture collections, WFCC it's called. It is launched in 1966. It is a global registry for culture collections and serves as an information resource for user community. So if you want to buy something, like if you want to purchase something from something online, you may be looking in Myntra and then Flipkart and Amazon or some other sites. So the importance of FCC is like, if you want to buy a microorganism, you can either look into individual websites, ATCC, MTCC website, or you can just go to FCC, WFCC, and just type. So it will be all the products in all these culture collections will be listed there. It's just like a database of everything, so you don't have to search everywhere. You just go and search in FCC. So things in different culture collections will be displayed in their website. So it's a registry for culture collections, and for users it is easy to get things and compare price and characteristics. It is a comprehensive directory for cultures, microorganism, and culture cell. It is not only for the microorganism, it's also for the animal and plant cells. It promotes and develop culture collections. It helps the give consultancy for starting of new culture collections. It will have an active network to ensure long-term perpetuation of important microbial culture collections. It's an organization which helps the growth of microbial culture collections. Thank you so much.